This is part 2 of our lecture on pharmacogenomics. And our objectives today is that at the end of this lecture, the student should be able to list the 5 factors that affects one's response to drugs, explain how genetic variability occurs and its use in pharmacogenomics, describe what single nucleotide polymorphisms are and their use in pharmacogenomics, and then we're going to look at some specific examples of how single nucleotide polymorphisms are used in pharmacogenomics. The factors that affect one's response to drugs are gender, age, size, habits, geography, race, genetics, and can make one responder or a non-responder. This is the central dogma of biology, and you can tell that at any one of these levels, you can generate genetic variability. For instance, at DNA replication, at transcription, or at translation. So, at DNA replication, you can have insertion, deletion, substitution, or rearrangement. At the messenger RNA level, you can have splicing in, splicing out of introns, or joining of exons that can be abnormal, or alternative splicing that will lead to different messenger RNA products and, of course, different proteins. And so, single nucleotide polymorphism mutations are very common in the human genome. There are over 10 million SNPs that are known, 120,000 of which are within exon. So there is an estimate of 4 SNPs per gene. SNPs can be exploited in drug discovery, and I've given you a website where you can go and look at some of the most common single nucleotide polymorphisms. This is a diagram or cartoon that we've looked at before, showing a test population in the center and a different subpopulation that show different responses to the drugs. And again, we know from our study, and we're going to find out, that this is based on single nucleotide polymorphism. And so when a person goes to the doctor, the person is given drug, and on my left, that person will develop toxicity to that drug, and so the physician will say, well, oops, that's too much. And so the physician would drop the dose, and then if it's still toxic, you drop the dose, and it could be that you drop the dose to the point where it has no effect. And then you have to increase the dose. On the other hand, individual goes to the physician, and they're given a drug, it has no effect. The physician will increase the dosage, no effect, increase the dosage, again, no effect, and so you just keep increasing it until you see some kind of beneficial effect. And so the point we're trying to make is this, that this is almost like a guesswork, because if you do not consider somebody's genetic variations, it's just all a guesswork. But if you look at somebody's single nucleotide polymorphism map, you will be able to determine which drugs they can be given and whether or not those drugs will be effective for their treatment. The enzyme system that's responsible for metabolizing 30% of the drugs that are on the market has over 90 variants. And this belongs to the big class of cytochrome